Now we are going to talk about the package ecosystem in RM Bioconductor that is available for microbiome studies. The package ecosystem provides different tools for analyzing microbial communities, different standardized tools. And these tools operate on the TreeSummarizedExperiment data container that we explained in the previous session. Microbial communities are complex dynamical systems, and we typically need various different analysis and modeling approaches to understand and analyze these communities. The package ecosystem provides different tools to share the various techniques that have been developed by the community of developers and to standardize these so that the rest of us can take advantage of them. These analysis tools are often rather complicated, and the packaging and standardization can help to cover the common use cases in a robust way. But we need to look at these packages and tools in the context of the overall analysis workflow. So the analysis workflow for microbiome data starts from importing the data into standardized data containers that can then provide the starting point for the downstream analysis of the community profiles. And in RM Bioconductor there is now an increasing ecosystem around the TreeSummarizedExperiment data container that binds together different types of information related to taxonomic profiling data. One of the main packages in this ecosystem is called MIA for microbiome analysis, but there are also other packages that we will go through today briefly. This is a collaborative effort between a number of different developers from different countries and institutions, and it was initially introduced formally in the European Bioconductor Conference 2 years ago. But the developments date back a longer time, of course. And the ecosystem is in constant development and expanding. So by bringing together these tools, we can complement the data presentation by providing various approaches, analysis approaches that take advantage of the data and support the community. And there are now different demonstration datasets available in a suitable format, and we can run them through the various tools. In the recent years, there has been rapidly increasing number of different R packages for the analysis of microbiome community data. My colleague Sudarshan Shetty has collected a survey of different tools for taxonomic profiling analysis in a website. And here are just some examples. There are many more. Many of these rely on a data container we call Philosec, but Philosec can be also converted into TreeSummarizedExperiment, and many of these tools are available for both communities. So the increasing number of microbiome analysis tools is also highlighting the need to standardize many of these things. To standardize the data representation formats and the need to standardize the way we are building the tools. And RM Bioconductor communities are providing good examples on that. Bioconductor is a global effort of standardizing many of the bioinformatics tools, and it started more than 20 years ago as a platform for microarray data analysis initially, and now there are more than 2,000 packages and several dozens of packages for microbiome analysis in particular. These tools are typically interoperable so that we can utilize tools built by different communities as long as we rely on similar data representation standards. And this is one of the strengths of the bioconductor ecosystem, that we are not only limited to the tools for analysis and visualization built by our own community, for instance microbiome researchers, but we can also utilize tools from other fields such as single-cell sequencing, which is using very similar techniques than we do in microbiome research in the context of R and bioconductor analysis. The microbiome data container was introduced in the previous session, and this is what we call TreeSummarizedExperiment. and it binds together the taxonomic abundance tables with side information on the taxonomic features and the samples. And it provides a standardized way of looking at the data, and the ecosystem of tools is able to take advantage of this data format. We can trust that the data is always provided in this particularly organized way, and this way it becomes more easy for the tools to understand how the data is structured and how it can be manipulated. This is not only specific for microbiome research, but in the R bioconductor ecosystem, all the different omics types utilize very similar approaches. There are also now example and demonstration datasets available in this format. So there are actually several different sources of demonstration data and open research data. So first of all, the R package ecosystem itself contains several demonstration datasets in the individual R packages, and these can be obtained through the standard data command. In addition, we are maintaining one microbiome datasets collection, which hosts several microbiome datasets in the TreeSummarizedExperiment format. But also other research teams have started to support this ecosystem of tools, and one prominent example is the curated metagenomic data project that some of you may have heard about. The curated metagenomic data is it's a huge effort to provide standardized and public datasets for microbiome data analysis. They have run all the different experiments from human microbiome studies through a standardized pipeline that processes the different datasets in a unified manner, and then provides the final output data, taxonomic abundance tables, and side information in the TreeSummarizedExperiment format. And you can have a look at more information from this publication that you see on the slide. There are now dozens of different human microbiome datasets available publicly using this standardized pipeline. Then there are the interoperable packages for analyzing such data. There are several different R packages that can be used for these purposes, and they provide techniques for data manipulation, visualization, and differential abundance analysis, and many other things. It is not only the packages that have been built for the microbiome analysis, but we can also utilize tools from RNA-seq, single-cell communities, and other communities that use similar tools and techniques. The collection of packages is not only a technical collection of packages, but it is also a community of users and developers that can come together online in online chat environments like Bioconductor Slack and Gitter Chat, and of course in live workshops, and we also have online tutorials available for the user community that supports the interoperability and collaborative use of these systems. So now we can have a closer look at some of these packages that are available for the microbiome analysis. So the MIA package is one of the key packages to analyze data that is provided in these standardized formats. And one of the key functions that it has is to provide import functions from several standardized formats where microbiome data is represented. So we have, for instance, the biome format, CHIME, two outputs, motor format, and other types of formats for microbiome studies, and we can import from these different formats to the TreeSummarizedExperiment data container. So this set of tools is a key for getting the data in R and Bioconductor in the desired format that is amenable for the downstream analysis. And once we get to that point and we have our data imported in R in the suitable format, we can utilize some other tools. And the MIA package provides tools, for instance, for the agglomeration of the taxonomic profiles to higher taxonomic levels, for splitting higher taxonomic levels to different abundance tables. We can transform the data. We can provide compositional transformations, CR transformations, other types of transformations. We can do manipulation for the data. For instance, we can calculate prevalences for the taxa and subset the data based on that. And we can do downstream analysis calculating alpha diversity indices for community diversity and beta diversity measures for measuring community similarity in terms of taxonomic composition, and many other types of analysis are available through the MIA package. These are all standard manipulation and analysis techniques that are common in microbiome research. The way we are using this is of course the command line operations in R, and here are just two examples of the types of commands that we can use using the MIA package. So one is the transform samples command that shows you how you can transform your data into the CLR format and add a pseudo count of 1. The other example shows how we can calculate phylogenetic aware distance metrics like unifrac, which is here provided in the weighted format in this example. In addition, there are other packages, for instance a package that is now in development for microbiome data simulation and provides different types of models, for instance Hubble's neutral model, generalized Lotka-Volterra model and self-organized instability model, to provide simulations of microbiome data. It's called miaSim. Microbiome time series analysis is also a common task, and the miaTime package provides some tools for the analysis of individual time series and their visualization, as well as community divergence analysis. The miaTime package complements the tools in MIA, which are for more standard manipulation without time series. And then of course we need to visualize the data in many use cases, and the miaViz package provides a set of tools for standard visualization tasks, starting from typical box plots and density plots, jitter plots, to principal coordinates analysis techniques and visualization of the tree information in microbial studies. So these are just some of the examples of the tools that are available in the ecosystem of analysis around the TreeSummarizedExperiment data object. By combining the data objects and these tools, we can start to build completely reproducible and transparent workflows for microbiome data science.